Hello, this is Mickey Urdea once again for Halteres Associates, and welcome to another episode of Interesting Stories in the History of Diagnostics. Today I'm going to continue the discussion about hepatitis C. Last time I ended with the work that had been done at Chiron Corporation in collaboration with Ortho on a blood screening assay for hepatitis C that was based on the detection of the antibodies. Now I'd like to talk about the explosion of activities that went on to carry on that type of work, not only in diagnostics, but also I would like to talk a little bit about therapeutics and also vaccines. With therapeutics, when we talked in the past about HIV, I mentioned the fact that we were able to culture HIV very early on. In fact, that was one of the ways in which it was possible to make enough of it to do the sequencing and a lot of other work. But we didn't have that luxury with hepatitis C. It wasn't culturable until many years after 1987. In fact, as I'll come back to later, it was in the 2000s. So you didn't have the luxury of doing what it was possible to do in HIV, which is actually culture the virus with potential antiviral agents. That wasn't possible to do. So what ended up happening is that a lot of work was done on drugs that were available for other purposes that people suspected were going to be useful for hepatitis C treatment. Two in particular are alpha interferon and then ribavirin. And that work was done over the course of the next many years. The approval of the first drug combination, matter of fact, which was alpha interferon and ribavirin, was in 1998. So, 11 years later. But a lot went on between the discovery and that, and I want to talk about that. And those drugs, by the way, were approved based on what was referred to as sustained viral response. And you didn't have the ability to look at the virus itself, so nucleic acids were the way in which that was done. And I'll talk much more about that. Vaccines are another issue. Still, although there have been a lot of reports about progress in vaccines, we still don't have a vaccine for hepatitis C. There was an intriguing report, though, that had to do with the potential of using an mRNA vaccine. Heard a lot about that recently, and it was used in a mouse model. It actually did involve in viral suppression within the mice. So maybe mRNA comes to the rescue again. We'll see, but we're not there yet. I ended last time talking about the use of detection of HCV antibodies, specifically for blood transfusion. But that then became used also for diagnosis of hepatitis C. And it's not really diagnosis of active infection. That's because many people actually resolve an HCV infection in 6 to 12 months. So they no longer have virus, probably, but they do have antibody. So if you detect positive antibody, it means you've been exposed before, but you might have cleared it. You don't know if they're active. That's why you then would test someone with an HCV RNA assay to determine whether there's actually presence of virus and therefore active infection. So I'm not going to come back to antibodies in the near term. I just wanted to indicate that there are issues with regard to the antibody alone for diagnosis. But I do want to spend quite a bit of time talking about the gain in understanding of hepatitis C variation and structure and how that became important both in diagnostics but also in therapy. As I've mentioned before, my team at Chiron was working on the branched DNA signal amplification technology. That's what we were using for HIV viral load. We had used it for HBV viral load, and now we wanted to use it for HCV viral load. But we got some lessons in some of those other applications that I want to talk about here. We realized from a previous experience how important understanding the variation in the sequence of a virus is to designing a diagnostic test. So specifically, I want to talk briefly about hepatitis B virus, which we'd done earlier. So we did that in the late 80s. We developed a quantitative viral load test for HBV. We did a lot of experiments with researchers in the United States and in Europe, and we had the desire to find funding to carry out the completion of that diagnostic development. So we knew that HBV was a big issue in Japan, and we thought that it would be useful to approach some Japanese companies and see if they were interested in helping us fund this and to work on it and get access to the product for Japan. As I mentioned, we did a lot of work in the US on this, and we said, well, why don't you send us some blinded samples? And we reached out to a number of companies. 3 companies decided that they would do that. They sent us blinded samples. We brought it back, and my head of research, Janice Kohlberg, and her team processed the samples, and we did some control studies on US samples. And at the end of that work, we sent the results back, and all 3 companies got back to us and said, you missed every single sample from Japan. although you got the US samples right. Our jaws just dropped, and we thought, what's going on here? We did know that there were some sequence variations between the common strains in the US and the common strains in Japan, but we just didn't think they were significant enough to get no signal in our test. But what we did was we redesigned the probes so they would take into consideration a Japanese strain too. We retested the samples, and they all were positive. The right ones were positive. But by then, those three companies said, "Forget it. This was too much work, and not even sure we believe the results." But we did manage to work with another company, Daiichi Pure Chemicals, and they eventually did fund the research going forward. But boy, did we learn the importance of using as much knowledge as you can possibly gain on sequence variation in these viruses before you try to develop these tests. So at the time that hepatitis C was cloned and sequenced, we then had, one sequence, kind of like at the end of the Human Genome Project, we had one human genome. Now what we have to do is look at the variations across the different strains of hepatitis C. So we started collecting a number of samples. We had some that were from George Kuo's team here at Chiron. We then obtained them from other researchers around the country and around the world. And we did an extensive sequencing analysis. A bit to our surprise, there was quite a bit of variation in the hepatitis C virus, and that led to our identification of at least 5 different strains around the world. In 1992, Tyan Cha, who was the key person in Janice Kohlberg's lab in my group at Chiron, wrote a paper that we published, and the title of the paper is "At least 5 related but distinct HCV genotypes exist." So that was published. We got a lot of good responses to that. But what we found out is in that same time frame, a whole lot of other people were doing exactly the same thing. They were trying to determine what the variation was in the hepatitis C genome. So this is now 5 years after the discovery of hepatitis C. And there are now international meetings on hepatitis C. And, I would go to these meetings. Some of my team would go to the meetings. And we'd all be there talking about these discoveries in hepatitis C. And different people would talk about these genotypes. But it was an alphabet soup. We had 4 different naming systems from different groups around the world. And people would refer to genotype 1 from this group, genotype A from this group, genotype X from this group. It was just completely crazy. The systems were not concordant. The systems overlapped, and sometimes there were extra genotypes here and not there. It was just crazy. And I thought it was an astonishing waste of time for us to go back and forth on this. We needed to have a way to have consensus. Janice and I took a look at the various publications that were out there from other groups on genotyping, and we agreed that someone in particular, a fellow named Peter Simmons at the University of Edinburgh in Scotland, had done the best job of acquiring and determining sequences and then putting them together in a very nice genetic map. And the nomenclature they came up with was actually really close to what we had come up with. So we reached out to him and said, we'd like to collaborate in this area. And we did. We published several papers together. But I was thinking about this issue of the problem of everybody having their own systems. And, we put our 2 together, but, everybody else didn't. So I thought a bit about how we might be able to get people to agree on this. And what I did was I reached out to the journal Hepatology, one of the key journals then and now. And one of the people on the editorial board who I knew, a fellow named Adrian Belli. And I said, Adrian, if we put together a paper for hepatology where we are able to gather a large number of the researchers in hepatitis C, and they all agree on a particular nomenclature, would you be willing to publish it? He said, sure, we'd love to do that. So I got together with Peter, and we actually wrote up a paper that we then sent out to a lot of the key people in the field, including Harvey Alter or Mike Houghton and his team, and a number of other people. And what our proposal is let's all agree to use this particular nomenclature. We try to take other people's work into consideration, but let's all rally around this. So I managed to get 45 people to put their names on the paper, put them all in alphabetical order, except that Peter was first author and I was last author. And from that point forward, once it was published, everybody rallied except for 2 groups, one from Japan and one from the United States, who just wouldn't sign. They wanted their particular method to be the one that was used, and it just didn't happen. So from that point forward, all the journals all over the world started using that one genotyping system. I've spent a fair amount of time talking about the background of genotyping, and you might just think, well, okay, that's interesting, but why does it matter? It mattered for two reasons. One I talked briefly about before, which was the ability to design the right probes and primers, depending on the technology that was being used for HCV tests. And in that regard, we learned a lot about where the sequence was conserved and where it's not. For instance, the most conserved region was the so-called 5' untranslated region of the virus, and then structural, non-structural proteins within the virus. It really made a big difference to have that understanding of the variation. The second reason is that it ended up that these genotypes don't all respond to drugs in the same way. And that became very important. And I'm going to go through that in a few moments here. But in terms of the testing, so we had viral load I'll come back to, but now there were genotyping tests that were being developed so that people could determine which genotype they had in a particular patient. And I think the best work done on that was from a company in Belgium called InnoGenetics. And I remember very fondly working with them and having many discussions with them. In fact, at one point, my head of business development, Tim Foltz, and I had a deal with the company so that we could, co-commercialize their genotyping and viral load. And we had a handshake with their CEO. Tim and I went away, and we found out the next morning that the CEO had gotten fired by the board, so the deal was off. But nevertheless, we had a lot of good relationship with them afterwards. And I remember one particular scientist there who I thought was really phenomenal, named Gert Mertens. Gert was a very interesting character. I got to know him relatively well, and he had a rock band called Red Zebra. And this was a CD I got from him. It's called A Red Zebra is Not a Dead Zebra. And there's a picture of, I'm sure you'll be able to recognize him on the street now. So that's him playing guitar for the band. So let me give you a little background on these genotypes, how many of them are, et cetera. There are 7 genotypes now known. You'll usually only see 6 listed because one of them, number 7, has only been found in one person. It was an immigrant from Africa to Canada, and that person had a very different sequence than all the other genotypes. That's the only one that's ever been reported to my knowledge. There are then so-called subtypes, and these subtypes are within each of the genotype classifications. There can be more than one subtype. For instance, genotype 1, subtype A, subtype B, or sometimes it's just called 1A, 1B. There are 2 genotype 1 subtypes, A and B I mentioned. There are 9 genotype 2 subtypes. There's only 1 genotype 3 subtype. There are 5 for genotype 4. There's 1 for genotype 5, and there are 2 for genotype 6. There's some interesting aspects to these genotypes, besides the response to drugs that I'll come to in a moment. But for instance, it ends up that genotype 1 and A are most common in the US, Western Europe, and Australia. About 6% of all of the strains there are one of those, A or B. In fact, in Japan, China, and Russia, it's mostly genotype 1B. In Southeast Asia, it's genotype 3 and 6 that are the most prevalent. One of the major transmission modes for hepatitis C is from the use of injectable drugs with dirty needles. And it ends up that all over the world, genotype 3 is the main form. And that's rather mysterious. And many people trace it back to perhaps in the Southeast Asian area, particularly back to the 60s and 70s when the U.S. military was there in Vietnam. So there's all kinds of information here about the spread of the virus throughout the world. And interestingly, in transfusions, it's mostly type 1. And now that's been going down as it's been controlled in transfusions with proper testing. So there's a lot to be learned here just in terms of the epidemiology and the history of the virus, but I'm not going to talk any more about that. It's just remarkably interesting. The greatest diversity of all of genotypes in a population is really in Africa. And I guess that's no surprise given that this is probably a relatively old disease, and that's the most diverse group of people on the planet. So you look at, for instance, Central Africa, and it's mostly genotype 4. If you look at West Africa, it's predominantly genotype 2. but you also find genotype 1 in some areas, particularly in Nigeria. You look at South Africa, and you find a lot of genotype 1, but in some areas, mostly genotype 2. In South Africa, a good portion of it is genotype 5. So you've got these very interesting differences in different places within Africa itself. And all 6 of the genotypes represented there, and also probably that one isolate of genotype 7 that I mentioned. While the combination of alpha-interferon and ribavirin was being studied, it became clear that the people with genotype 1 tended to respond more poorly than genotype 2. It ended up later, as I'll talk about, that the viral load in type 2 tends to be lower than in type 1. Also, they found that people with a lower viral load in type 1 responded better than people with a higher load in type 1. So there's some of the amount as well as the genotype involved here. But that was some of the early work. So that's using those first 2 drugs. But in 2005, a Japanese group actually was able to take a molecular clone of HCV and express it in cell culture. That was a big change. So now we suddenly have the kind of tool that we had back in the HIV days, where you could propagate this in culture and you could start to develop antiviral drugs in the classic way. And so that led to some of the very first antiviral drugs, small molecules that were designed specifically to combat HCV infection. And the first of those was sofosbuvir. These antiviral drugs became collectively known as direct-acting antivirals, or DAAs. And you'll hear that a lot in the literature, and oftentimes people will talk about availability of DAAs for treatment of HCV. As more and more of these small drugs were designed for HCV, it became crazier and crazier, because it got to a point where there were approximately 12 different drugs, and the use of them was usually in combination, different combinations for different purposes. So in fact, there were different combinations that would be used for different genotypes, and also for whether someone did or didn't have cirrhosis, whether or not there was presence of any antiviral mutations against some of the drugs that cause resistance, and then different regimens of either 8 weeks, 12 weeks, or 24 weeks, sometimes even 48 weeks of treatment. And that would be dependent on the genotypes. And you look at these recommendations, these guidelines, it would have, you need a fairly substantial wall chart for someone to use. So people were rather hesitant to use these combinations. It was very complicated. It wasn't so simple. You need to have a fairly substantial formulary of different drugs and different ways in which you're going to use them that depended on the genotype that was in your particular location. I'm going to come back to where we've gone since then. And that wasn't that long ago. That was, 5 or 6 years ago even. But things have changed a lot. And spoiler alert is it's gotten way easier. So I spoke a lot about one major aspect of nucleic acids in hepatitis C, which is sequence variation, how important that was for designing probes and primers, but also then the identification of different genotypes and subtypes, and how that became important in understanding who will and won't respond, and how to select particular drug regimens for particular people. The next thing I want to talk about in the next episode will be viral load in much more detail. Talk a bit about some of the work that was being done simultaneously on viral load, as well as the genotyping, and how it was being used. It was initially used the way I expected, which had to do with monitoring how someone responds to interferon and some of the DAAs, but also then the likelihood a particular individual would actually respond to these drugs or not. That became important as well. So, and then we were able to use it to look at a lot of other things, such as where the virus occurs, whether it's different in different disease states, and how that could be used to understand how best to manage a patient. So I'll go through those things in detail on the next episode. So thank you very much for listening to this episode. This is Mickey Urdea for Halteres Associates, and I look forward to talking with you next time. Goodbye.